So that's, the nice building and this nice picture that's here is actually one of our buildings of the Institute. And not the building where the mass specs are, but, it's a really good picture and I like it. It's because it really looks really inviting there. So before we start, let me just acknowledge the country, and Griffith University acknowledges the people who are the traditional custodians of the land. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander people. So where are we? We are actually in Australia, and for the ones that don't exactly know where the Gold Coast lies, it's essentially just here, pretty much on the most eastern part of Australia, on the east coast, just about an hour south of Brisbane. And here it's a beautiful place to live in. That's pretty much where our institute is located, down the track in here. It's about 15 minutes away from the beach, and that beautiful beachfront on here is essentially what we call the Gold Coast, the beautiful Gold Coast. And it's essentially not that far away from the US. It's just roughly around 1,600 kilometers south west, essentially, from San Diego. So that's just up there. Alrighty. So what we are doing in my lab is, on one hand, we are working on tools and how we can essentially integrate glycomics, glycoproteomics, and other aspects essentially of information. Sorry for the police kind of back. In on understanding what glycosylation actually can do, and of course we apply these. The two major areas of biology that we are looking and working in is essentially in the cancer and stem cell signaling field. And I'm going to really talk a little bit more about the stem cell aspect today. But also one aspect that came up in the last couple of years where we have been trying to figure out a little bit more is more of understanding what glycosylation does across animals, especially in vertebrae and chordata in particular. And also how this essentially can spin into the context of zoonotic transmission. And these are the two topics that I'm going to talk today a little. So first of all is really the Glycomics aspect and what we can do around that. That's what we're going to get started with. Okay, so we all know what, I think most of us in this audience is going to agree what evolution is and where we are coming from in one or the other way. And there are a ton of different type of living beings on this world, from very comparably simple monocellular beings up to the multicellular beings that essentially make up the vertebrae. And while we have, in the glycobiology context, have gotten a much better understanding that glycosylation is a very universal aspect across pretty much every life form that we know nowadays. It's especially in the field of the vertebrae where we actually really, I think, missing a lot of information and knowledge from the systematic point of view. What's the diversity of glycosylation that we actually have in this particular field? So we know plants, we know insects, we know fungi, we know this in this big picture on there, but how does it actually look within our small little, evolutionary niche there? That's something that if you start looking at a little bit more detail, you actually don't find too much of that. And I don't have to introduce too much about this essentially in this audience here today. We know that the basic features that plants and insects, slime molds or yeast and fungi have in the first place essentially are quite distinct and well known. And for vertebrae, we usually assume, okay, we have this core trisaccharide, core pentasaccharide there. We have core fucosylation, then extension of the arms, and then different variations of silation, fucosylation, modifications of these arms in one or the other way on the complex type glycans. So that's pretty much what we think we know, and this is just to make it a long story short, there is actually not wrong at all. But how are these different glycan features that can be there are actually being used across the different type of animals, from lungfish and bony fish essentially up to to the living beings or that developed comparably recently on there is actually not really well known in a systematic manner. What you can find is one or the other strange protein that somebody purified from that animal somehow or from a different animal. But done with a variety of different methodologies. And what this essentially limits us is actually really getting a top-level comparison view across these species. So that's a little bit, where we are coming from to actually see how we can change this. Okay, so why, of course, and again, I think I can keep this short for this particular audience, is because glycosylation and glycoconjugates are just intrinsically involved in a lot of interactions, whether it's essentially within their own cellular communication processes, whether it's for transmission and infection purposes, but also in the context of food allergies. And so it's an absolute critical feature of living beings. And that's why we are actually trying to look at this in more detail. Okay, so what we did in this particular project, we started trying to collect as many sera as we could actually get from as many fish, or let's say as many animals across the major evolutionary clades that are known, as as much as we could actually get. And the reason why we took serum for that in the first place is just because it kind of fulfills the same function across all animals, and it's the easiest one to get that is also comparably non-invasive and doesn't require to have to kill any animals in the first place. So just from a purely ethical point of view and organizational point of view, it was really the only option. And if you start looking from what we understand, when certain of the animals that we actually had in our study, so all of these animals that are listed in here are animals that we had in our study, when they or when we think they started to evolve across the last 400 million of years, you see that there's quite a long distribution of the different animals from around 400 million years before and until very recently in the last 100 to 50 million years on there. Okay, why do we want to do phylogenomics for that matter? If phylogenetics already did everything, we can get all the sequences out, we can see how these animals align, how closely related the sequences are. But what these sequences don't tell us and don't give us is actually how the different transferases and glycomodifying enzymes are actually being used in an animal. So yes, we know that pretty much all of the animals that we know that are out there have some form of food aid, the core fucosyl-transferase. All of them have MGAT1. All of them have, in one or the other way, cytokines or homologs of them in one or the other way. But it doesn't really tell us anything about how they're being used. So looking at the glycom, at least, it allows us to actually really get a much better idea about the repertoire and the ability of an animal to use glycans in one particular way. And I understand the limitation of this study is here. We're just looking at serum. We're not looking at other organs. We're not looking at other aspects that would be critical. But you need to start somewhere. And second, it was just not feasible purely from a technical and from a feasibility point of view. Okay. I already kind of mentioned this. We are wanting to look at the sugars and the glycoconjugates there in the first place because what we can deduct from genes and proteins that we can get out of that are the type of tools that are available, but it doesn't tell us about anything about what substrate specificities are there. It doesn't tell us about the transferase expression levels. It doesn't tell us about what substrates might be available in certain animals. And also, last but not least, about the activity of the transferases. So Glycomics and looking at the glycans for that particular matter allows us to actually look at this in a combined fashion. We might not be always able to get everything out of that, but we also found earlier that there is not always a direct connect between what type of transferases are expressed or changes of them that they do not necessarily reflect in the glycome profile in one or the other way. And that's why we're really trying to do this from the glycomics point of view. And that gets me to the point that I actually need to introduce, a former PhD student of mine, Dr. Bana Murugan, so obviously she finished, and who was actually doing the majority of the work really in this amazing study there, where we really took plasma and serum from a couple of different animals. We also looked at the immunoglobulins in a separate manner, but I'm not going to talk about the immunoglobulins today. and really used our PGC approach in the first part to really understand a little bit more about what features are present in which type of animals. That's just kind of, a list of all of the animals that we actually had. We usually try to get at least 3 different animals per species. Was not possible for all of them, but for most of them we have at least 3, if not more. And some of them are even on endangered, vulnerable, or nearly threatened list of animals as well. So maybe the information that we can gather here will help eventually some of them as well in a different context. This, however, would just not be possible, however, with an amazing set of collaborators that we were able to gather, people and colleagues where we were able to get samples from Australia as well as internationally. We also bought a lot of zero on there, but also with people on a more computational side that are helping us to actually now get this data together in a way that it's going to be available as well in the very near future in there. So I'm not going to name everything by name in here, but all of them had a critical aspect and contribution in this particular project to make this happen. So before we go to some of the results, I just want to quickly brush over how we actually do our Glycomics approach on here. So, and that's an approach that pretty much has been developed in Nikki's lab some time ago, which has been further modified and is being still further developed in order to become better. In this case, what we are doing is we are immobilizing our glycoproteins on a PVDF membrane and can use essentially PNGSF to release the N-glycans in the first step, followed in the second step by reductive beta elimination from the very same sample, which allows us essentially to get N-glycans and O-glycans from each and every sample that we're looking at. And the reason why we're using PGC-LC for that is really just because we're getting, with a single type of analysis, a lot of information. Why? So, for example, rather than just get a compositional idea about, for example, in this case, from some short-nosed fruit bat, where the 2 major structures are pretty much a diantennary-sialylated glycan with 1 or 2 sialic acids. We can actually get a lot of structural information out of the fact that the retention time that we can determine is essentially quite unique on the PGC level, but we can also essentially get a quantitation out of that. So you take the respective chromatograms on there to get a relative quantitation of the different structures of the presence. We can use the retention time to actually separate different glycan isomers that otherwise would be isobaric, to see what level of 2,6 or 2,3 linked sialic acids are present in this particular type of sample. We get the MS2 spectra for all of the glycans as well that we can use to get further information and details about the structure. And together it's just essentially a very powerful tool out of the many other powerful tools that are out there to are doing Glycomics. But in our case, it just works very well to get a lot of this information done in a single go. And why I like to use our approach in that particular context is because, especially in a project like this, where we have been embracing a couple of interesting surprises down the track in terms of what we can find in the glycans. It's just one example in here where we get, one major glycan. In that case, it's essentially doubly negatively charged glycan of 1046.8, which pretty much could be 2 different compositions as indicated in here. So it could be either a glycan with an alpha-gal modification and then an acetylneuramidic acid, or it could be a singly sialylated glycan with an glycosylaneuramidic acid and the core fucose, the Lewis fucose somewhere around on there. And that essentially is not always an easy to determine just purely from a compositional level, but this is where our MS2 approach actually can provide us with a lot of information. And you can see already here what we can actually find. Here is that, there's one arm containing alpha-galactose residues, another arm containing the sialic acid there. And that essentially provides us with a lot of the information that we actually need to get. In comparison to that, also the glycosylated version of that looks slightly different, but has the same type of features. And, without taking too much of what is going to come in there as well, we also saw that the arm distribution, for example, was something that in a couple of the animals was quite specific. So the alpha-gal was essentially exclusively found just on one arm, whereas the silation was found on the other arm, which of course poses a lot of other interesting questions down the track. How do the transferases control this type of specificity in one or the other way? That's not a question I'm going to have any answers to, but this is at least, some of the type of questions that we can actually extract from the type of data that we can get. Another example for why I really enjoy our particular approach for this project in particular is, when we're just looking here in that case of two different bed types in one or the other way, which, pretty much look very, very similar, having pretty much the same type of composition of a glycan in there. However, in the PGC, we can immediately see that there's actually not just the same glycan in there, because whereas essentially one of the beds essentially has a very 2,6 focused silation of its diantennary glycan, and other one essentially has no 2,6 silated glycans, and uses essentially 2,3 silated glycans on there. So this is something that in our PGC approach comes immediately out in a single experiment, without having actually to go into additional aspects of exoglycosidase digestion to figure these details out. Okay. Another interesting finding was, for example, Lungfish, which Evolutionary actually, is quite early on, which, contains a lot of the oligomannose type precursors that you would expect to see from a eukaryotic organism in the first place. It's shown essentially with the green bars in here. But you can see there's also a couple of other structures in there that also nicely separate by hexoses essentially there, but that do not fit into this oligomannose type feature on there. So what it turns out to be actually in that case is, some kind of weird diantennary glycans that actually just contain some alpha and some beta galactoses essentially there. And that was, pretty much confirmed with exoglycosidases, which we use a lot, especially in some of these animals on that, to get a little bit more structural information on that by itself, by the PGC, are not easily determinable without the accurate standards in the first place. So when using an alpha galactose, you can immediately see how two galactoses are being cut down in here. Beta galactosidase didn't change anything on these major one glycans in there. So that also allows us then to provide a little bit more structural details and information that otherwise would be more difficult to actually get. So this is one of the major glycans in Lungfish. So when I'm not going to go now to each and every species essentially on there. So I'm going to go now down a little bit more to the top level summary in here. So when we now start looking at overall of all the glycans that we could find across the different species in here, one thing that became quite clear across, Serum is that pretty much every animal has their 2, 3, 4 major type of glycans that essentially make the bulk of the serum N-glycom in particular on there, with a lot of other small intensity glycans that are present across, the other ones. So pretty much taking the, let's say, 10, 11 most abundant type of glycans in each and every type of animal, you can very quickly get a good idea about what type of, animal that actually is. We started looking at this in a little bit more detail, of course, and that's what you can see here, and what are the top major glycans that you can find in most of the animals. And turns out that a lot of animals actually decided to really go down this diantennary disylated route as the major plasma glycoproteins in the first place. By far not all, and the intensities are actually quite different. Then we have essentially a whole set of animals that actually have the same type of glycan, but exclusively into 3 sialic acids. We have some animals that actually prefer the same, but with exclusively N-glycosylaminic acids, and that essentially are especially any bovines, mice, and some monkeys essentially on that really have a strong preference for new GC rather than new AC. And then there are some animals that actually have a very high level of co-glycosylation, like certain pigs and certain turtles, essentially, that they really do this. So by starting to essentially, pull out, some of the major features essentially on them, we can get a fairly good idea what must be the transferases behind that. Unfortunately, for a lot of the animals in the study, there would not be any genetic sequences available, so we can't actually go back down in the way to actually get a better idea from the protein structure point of view of the transferases. But on the other hand, it also provides us with an amazing opportunity to actually, in a non-protein biased manner, so to say, to actually establish how close is the glycom related, essentially. When looking at silation, and you might have seen it already beforehand in essentially the other slide. So we also did, released sialic acid, so just doing DMB labeling to get a better idea about the level of acetylation that might or might not be there, and the type of sialic acids and the absolute levels. And interestingly enough, as I mentioned already, most of the animals that we had, and that didn't really make a difference from an evolutionary point of view, preferred NAC as the major sialic acid in plasma. That doesn't mean that many of these animals cannot produce NGC. They have NGC in single-digit percent levels available, but they just on purposely do not seem to choose NGC for their plasma glycoproteins in the first place, with the exception of some mice, monkeys, and of course beef. Acetylation was not such a strong feature in most animals. There's some higher acetylation, like in echidna, for example, in possums or some of our Australian animals there. Some rats and horses had higher levels of acetylation that we usually wouldn't see in the PGC approach. But yeah, so in terms of that, the serum has still a lot to offer in terms of different features of glycosylation. We also had the possibility to really look at a couple of different mice strains. And funnily enough, between mice and rats, the major difference really is the use of the sialic acid, where rats really very strongly prefer NAC rather than NGC. But also between the different strains, the glycol itself was essentially showed slight tendencies to be strain specific. I wouldn't say it's an absolute one, but there were definitely differences between the strains that were detectable. But just looking at this data, the one thing that I can't get rid of thinking is actually that probably rats might, for a lot of glycosylation-related questions, be the much better model organisms compared to mice, at least with respect to the proximity to humans, just given the fact that their preferred type of sialic acid for a lot of protein seems to be N-acetyl-muraminic acid rather than UGC. Okay, so this is something that we are currently putting together. So Urbana is doing a great job in getting this finalized and finished. So stay tuned. This is going to hopefully be available quite soon. And yeah, out of that, really one major question that comes out of that, and I'll not going to have an answer for that at this point in time, but I think we have sufficient data to actually ask ourselves the question to see whether there might actually be the evolution on the level of glycosylation might actually not be entirely dependent on the level of how the genes are evolving or how the proteins are evolving, but much more how the animals choose to actually use these tools to make their glycans. Why I'm saying this? So when we look at the data that we had, we found, for example, very similar glycosylation features that were close to what we would say for human glycosylation in certain type of lizards and certain type of frogs, or for example, birds like chicken. While other birds like ducks, for example, or other Australian birds on there had very different features of glycosylation, from like Dynex essentially on there as the major glycans in there. So there is there's, and these are animals that actually have evolved away from our traditional, human clade a long, long time ago, but that on a glycosylation level, we're much, much closer, at least for serum, to related to humans than they are related to, for example, monkeys in one or the other way, which are our closest living relatives for obvious reasons. So I think it's not completely impossible to believe that actually that glycosylation and the evolution of glycosylation might be very strongly dependent on a lot of environmental and other evolutionary pressures that might not kind of go down back to changes on the genome level, but might actually go back down to changes on the way how the glycoconjugates are being built together. So that's our current theory. How it's going to work out, we're going to see. But yeah, I think it's reasonable to say from this type of data that there might be a much bigger role in the way of glycosylation is evolving than what we think. And that's where Glycomics really comes into place, because it provides us with this top-down level of information of how the combination of genes and proteins of the glycosylation machinery in an animal are actually being used to give you a picture of the glycosylation capacity. So I think that that Phylogenomics can complement phylogenetics in a very good way in terms of understanding what is happening across species. Okay, with that, let's go to the second part of the talk, which changing gears completely into a completely different field, and that is really the role of glycosylation in the context of ex vivo erythropoiesis. And before I start with that, I'll really just have to put out the people that actually do most of the work in there, especially Pauline, who has been pretty much being single-handed in putting most of the information together that you see in the Glycomics and Proteomics part. Becky, who is our expert at the Red Cross to really do the ex vivo erythropoiesis and controlling that part. And Larissa and Alpesh, who are amazing collaborators on the structure biology and molecular dynamics point of view to understand what's going on there. Okay, in this part of the story, I'm going to have to give you a quick introduction to stem cell factor and its receptor c-Kit. A little bit why we're using this ex vivo erythropoiesis model system on there before we dunk into the results that we have so far in the topic. Okay, so stem cell factor, or SCF, as I'm going to call it most of the time in the next couple of minutes, is a growth factor that is regulating the activation of c-Kit, or also called CD117, of this receptor. So both of them play a fundamental important role in hematopoiesis, in melangenesis, in gametogenesis, and mast cell regulation. And both a knockout of SCF of c-Kit in mice is embryonically lethal. So both of these proteins by themselves are absolutely critical for proper development of a multicellular vertebrate organism, as far as we know. Both of them are actually heavily glycosylated, and you kind of have this indicated in the model in here, where you see the blue and the teal chain being essentially the extracellular domain of c-Kit, the receptor, which itself is heavily glycosylated, and in the middle, the dimer of stem cell factor that sits between these two. So not just that they're heavily glycosylated, but the glycosylation is also highly conserved. And this is just kind of giving you a quick overview for c-Kit, for the receptor, with its different sites of glycosylation, and all the green letters there essentially indicate glycosylation sites and O-Glycosylation sites that actually are being used, whereas the red one would be essentially potential glycosylation sites, or the same homologous sequence there, but is not glycosylated either due to a change in the N-glycosylation sequence or an exchange of the asparagine into a different amino acid. For stem cell factor itself, which is also expressed initially as a transmembrane protein in there, all the known and predicted glycosylation sites are across a lot of animals that we know and have sequences for. These sites are extremely highly conserved. So well, I might be biased with respect to glycosylation on there, but that kind of tells me that there must be some more important role to glycosylation to it than just it's present itself. Bear with me, it's not too difficult what comes now. It's just kind of a need to give you a little bit of a background what the dynamics and what the diversity of these two proteins actually is in vivo, and why this is an interesting topic to go into. So we're looking essentially at a stem cell factor itself, which is initially expressed as a transmembrane protein on there, which has a couple of glycosylation sites, which can be not all fully occupied. And an O-Glycosylation site, you have a membrane-bound form, which essentially will be membrane-bound mostly, but you also have a cleavage site on there, which essentially releases a soluble form of SCF. And both of them, both the soluble and the transmembrane-bound form, are critical and important and fulfill, as far as we know, different functions within the body. We know that not all sites of glycosylation are always fully occupied, so we have some heterogeneity in terms of the site occupancy for the N-glycosylation, in particular on stem cell factor. And we also have essentially a mouse model, which is called the steel-dicky mouse, where no soluble stem cell, no transmembrane bound stem cell factor can be produced. So just a soluble version of that can be produced due to a mutation in the sequence there. And what these mice essentially experience is they are quite depleted in hematopoietic stem cells and progenitor cells. They suffer from severe deficiencies in pigmentation, as well as germ and blood cells, but at least they're alive. So this kind of really tells us there's a lot of different functions associated to this particular pair of cytokine receptor pair. However, what also happens is that, most stem cell labs in the world will pretty much use E. coli-produced stem cell factor, because it's one of the glycoproteins that can be easily expressed in E. coli and still essentially be refolded into a functional and active form of the protein. So naturally, this product is much cheaper than any of the stem cell factor produced in eukaryotic cells, but it's essentially a form of stem cell factor that does not occur, to the best of our knowledge, in any human living being. Okay, so these different proteoforms, we know that they fulfill distinct functional roles, but in terms of the glycosylation, the role that glycosylation might actually play there, we don't really know a lot. And this is, why we kind of got into this particular project. In order to actually now understand what is happening on the biological level on there, we needed a good model system. And ex vivo erythropoiesis kind of offered itself as a perfect system for that purpose for a variety of reasons. First of all, the majority of human cells, just purely in terms of number, are erythrocytes in the first place. So we're talking about a very important and highly abundant cell type in the human body, which is, producing, and every day, every hour, being produced a lot of these cells endogenously each day. SCF is an absolute essential factor for this. And given the fact that the supply of red blood cells remains a challenge, especially for rare blood group types, and I'm not talking necessarily about the ABO itself here, but more of the rare blood group types on there, there need to be strategies out there where we can probably be able to supply blood for these rare blood group types that does not necessarily rely on donors. And that's why we actually, understanding how this works, how we can use the knowledge of erythropoiesis and the role that glycosylation plays there, might actually also help us to produce better precision blood supplies in the future. So in this model system, which essentially is, starting from CD34 positive and hematopoietic stem cells that are enriched from peripheral blood from blood donations. So there's essentially a two-stage culture system that is being employed in order to essentially build reticulocytes there. And that's also one reason why I really like this particular system, is that, it's really as close as it can be to a human system, given the fact that the cells are coming from human donations. And we also can capture really the variety of biology in that particular system. And Becky, who is, one of our collaborators in this project, is one of the people that actually have been quite leading in developing this ex vivo erythropoiesis system on there. So what happens in this two-stage culture system is that in stage one, cytokines like stem cell factor are actively added in this in the culture to actually get the cells ready for production, get the cells ready for differentiation towards the reticulocytes in the end of the day. And in the second stage, stem cell factor is actually removed from the system. So we have two distinct stages, one where stem cell factor is being used, and the second one where no stem cell factor is actually required anymore in this particular system. Keep that in mind. And the key questions that we actually had in here was, okay, first of all, does stem cell factor glycosylation have some measurable effects on this particular system? Then if that is the case, how does this affect the interaction with c-Kit? And also what else is going on a more system-wide level going on in these cells, in terms of the proteome or the glycome in this particular context. And, and if all of that is happening in one or the other way, why? Okay, so what we did, we took our CD34 positive stem cells, had started the entire culture system at different levels of stem cell factor, and we're using glycosylated HEK produced one as well as E. coli produced one, which is not glycosylated, and then essentially observe what is happening in the expansion and morphology in the culture system in the first place. And in the second step, we also, of course, took at different points in this entire workflow. We took the different cells. We looked at the differentiation with flow cytometry. We looked at them with classical label-free bottom-up proteomics and an O-Glycomics in here, and then also figuring out what's going on. And that at 4 different time points at day 7, 10, 14, and 18 of the culture. So when we're looking at cell expansion and morphology, so what we can see in the first place is that in terms of the overall viability on there doesn't really seem to be too much of a difference between the glycosylated and non-glycosylated version. And the glycosylated version starts off to get be finished a little bit earlier, but this makes sense as you will see later on there. But otherwise, that's pretty much comparable. However, when we start looking at the fold expansion levels, especially at the point in time when it becomes relevant for these cells to actually develop into reticulocytes on there, we find a significant increase in the fold expansion levels, especially for the 10 nanogram version, and not so much for the 50 nanogram version anymore, but that makes sense biologically, as you will see a little bit later on. And so that already was quite surprising to see in the first place. But where this actually makes a huge effect in the end of the day is really in the amount of erythrocytes that you can actually get in the end of the day. So using a glycosylated stem cell factor, actually the amount of cells that you can produce from the same amount of stem cells that you start with is about 4 to 5 times more compared to the version when you use a non-glycosylated stem cell factor. Looking at certain differentiation rates essentially on there at different days with flow cytometry, they were essentially quite comparable, whether we're looking at beta integrins or alpha integrins or GYPA there, that looks very much in the way as you would expect it to see for the cells at this point in time of the development, with essentially a strong reduction of the integrins and a pretty much unchanged level of GYPA. When we looked at the proteomics part on there, we overall didn't really find, from the qualitative point of view, any particular difference between the different sets. I mean, there were just some few proteins that were just detected in one or the other type of sample, but overall, most proteins were essentially detected across all of them. And what also essentially our data confirmed is what has been already reported earlier, that during erythropoiesis, the number of proteins, of course, is going to be reduced just purely because what happens is that reticulocytes pretty much get rid of pretty much everything in the cell of the cell production machinery. Anything that is not related to capturing oxygen, transporting oxygen, essentially on there is being pretty much kicked out. And that's what you would expect to see from erythrocyte ghosts pretty much at the end of the day. So that looks also fairly normal. However, when we started looking at it from the quantitative point of view, it became very obvious that non-glycosylated stem cell factor regulates completely different proteins in a different way compared to using glycosylated stem cell factor as a cytokine in here. And in the PCA plot, you can very clearly see how the glycosylated versions, independent of the level, actually, produced a completely different proteome profile compared to the non-glycosylated one. And you're looking at this in a little bit more detail. So out of the roughly 1,200 proteins that were actually detected across all of them, approximately a quarter of proteins were completely differently regulated. And this is essentially were completely differently regulated. And that already indicated there's much more going on compared to what we would essentially expect to see if glycosylation doesn't actually play a role. So especially in the early stages, that's essentially at day 7, so where stem cell factor is still part of the culture on there. There's a lot going on in terms of the regulation. What is going on? So especially glycosylated stem cell factor regulates, upregulates proteins that are important for translation, for gene expression, for a lot of the cellular metabolic processes that are actually happening, for RNA splicing, for mRNA processing, for spliceosomal complex assembly. So a lot of proteins that are actually relevant to produce proteins to get the cells essentially to produce erythrocytes or reticulocytes in the first place, which kind of makes sense given the fact that we see so many more being produced out of the same type of culture. When we look at day 10, what are the top terms that are being increased? And you can see here again the huge difference between the human version, so the glycosylated version versus the non-glycosylated version, especially at day 10, in terms of RNA binding, cellular respiration, oxyreductase activity, etc. So there's a lot of things going on in this particular context that all indicate there's production, whereas essentially for the non-glycosylated version, a lot of, let's say, danger signals and a lot of degradation pathways are actually being turned on for reasons that we don't know yet. So also what it means is that with very low concentrations of the glycosylated ones, we can actually achieve equal to not better numbers of cells compared to the non-glycosylated one. So, of course, naturally, we know that the proteome changes across erythropoiesis, and that kind of really is reflected in here. But the reason why I actually have this in there is you can really very nicely see that, especially in the first stages of the culture, so when SCF is still present, so at day 7, at day 10, we get a very clear separation between the cells that are stimulated with glycosylated stem cell factor, which starts to become a little bit more blurred towards the point when stem cell factor is not in the system anymore. But even there, we still get a fairly good clustering between the cells, whether they have been stimulated with stem cell factor or glycosylated one or not, which indicates that there's still quite a tremendous memory response in these cells on the way of essentially developing into reticulocytes out of there, even though stem cell factor is itself not in the culture at all anymore. So that's just the number of proteins that actually goes down. When looking at the glycom, and this is for the N glycom on there, we see two different pictures. First of all, a dependence on what type of stem cell factor has been used, which kind of becomes a little bit less clear down the track, but also essentially that there is a change in the overall glycome across the different points of development, which is something that, to the best of our knowledge, has not been really looked at as part of the erythropoiesis, so to say. So there is a lot of glycosylation changes going on in that particular case in this time frame. And starting to look at what type of features are changing and how are they changing, we see certain hybrid type structures or hybrid type structures going a little bit up with the higher rates of the stem cell factor. So this is a fairly complex slide on there, but really to keep the take-home message on there are slight changes of glycosylation happening then in both in hybrid and complex type in particular, also a little bit in the oligomannose, even especially the oligomannose type glycans. And if you kind of put this into numbers in a much easier way to look at, so we really see an increase in complex type glycans from around 75 to around 90% in the major erythropoiesis, whereas oligomannose type glycans go down from around 23 to 8% over the time period, whereas paucimannose and hybrid type glycans are pretty much staying the same in the very low percentage range across this time period of development. When looking at some individual structures on there, so you see that some individual structures seem to be changing quite significantly, but more depending on the level of stem cell factor rather than the type of stem cell factor that is being used. So this is something that going to keep us busy for some time to figure that out a little bit more detail. But structures across all type of glycans are being affected by that. And when we now start looking at the glycome in a more holistic manner in that context in here, the one thing that really stood out in this PCA plot is that while the levels of the higher levels of stem cell factor seem to be clustering together between the E. coli and the human one on there, as you already indicated on there, when stimulating with a lower amount of stem cell factor on there, these cells are still having a quite distinct glycome between the non-glycosylated and glycosylated stem cell factor stimulation in the first place. So at this level, which physiologically might be even more relevant on there, that might actually be a much more impact on the glycosylation that we have there. And it's just interesting to see that there's a memory effect, even though the last 8 days of the culture, there's no stem cell factor in there anymore. So how do we explain this? We're still on the way of figuring out a lot of these questions in here. But one of the major points that we can actually start putting in there is that glycosylation must have some form and role in terms of the way how stable the interaction is. And we did this together with Alpesh, who simulated a little bit on the molecular dynamics aspect with no glycosylation and with glycosylation on there. And I'm not quite sure how good it's going to be able to see this on the video on there, but what the data from the simulations indicate is that glycosylation actually actively contributes to the stabilization of the stem cell factor c-KIT complex, so giving it a much tighter binding, giving it a much stronger signaling, and that would fit to what we are seeing on the proteomics point of view, where we see that the lower level of stem cell factor seems to be not having the same tight binding, and in consequence not being able to essentially elicit the same stable signaling that we actually get with the glycosylated version, and that this is essentially counter, or to a certain extent at least, counteracted by just using a higher level of stem cell factor, that that the signaling gets a little bit more stable, but you still see differences in there. So just a higher level of stem cell factor itself doesn't solve the problem entirely. So with that, I hope I could give you a little bit of an idea of the projects that are going on in my lab at the moment. I don't have to convince you that glycosylation is not just a colorful add-on, but that it's actually quite important. That also key signaling pathways can actually be differentially regulated as a consequence of glycoprotein interactions. So that really understanding of how glycans actually contribute to the interaction of such a cytokine receptor interaction might actually be really, really, really important to understand what is happening downstream as well. So that cytokine glycosylation obviously is probably much more important than what we think of. With that, of course, we have a whole ton of questions that we are trying to solve now, and thanks to the ARC, we can actually do this now. So stay tuned. There's more to come in the coming years on this particular project. And with that, I really have to thank my team that actually did all of this work in here. So Abana and Pauline, especially, the two major drivers for the two projects that I have essentially shown here today. Also our collaboration partners and funding agencies on there. If somebody is interested to actually, jump into that, we have postdoc opportunities available in this particular range. So, feel free to contact me. And I can't stop without actually mentioning two of the many glyco events that are coming up next year. But, one, of course, the GSC down in Italy in March, but also the omics conference in May in Cairns in Australia, which is actually a conference that is bringing together the Australian Proteomics, the Mass Spec Society, the Metabolomics, and the Glycobiology Society of Australia.